Hello guys, it's ready with another video which I'm going to talk about today. It is going to be structure of the antibodies. Nowadays, antibodies, a lot of discussion going on that the vaccines are activating the antibodies. But before we understand how this antibodies are going to be activated and how they're going to function, it would be very difficult to commemorate the fact how this antibody functions. Let's go into the detail of the antibodies. There we'll talk about how exactly the antibodies are going to work. I'll share with you about the antibody structure. If you see this diagram, this suggests that uh, the antibody structure, which is a glycoprotein that is made by the B cell, and once the B cell get differentiated into the plasma cell, it is going to make you the antibodies. So antibodies, they're going to be secretory in nature. If you have a little bit information about the cell biology, the antibodies can be made to sit on the surface of the B cell, and if the certain changes occur, it could be secreted out. That is another part of the you know the cell biology. But let's focus today on the antibodies structure strictly. So first, let me talk about there are certain receptors which in the form of the antibodies found on the surface of the B cell. And these antibodies, it varies, as for the class switching occur. And this is going to be specific for a particular antigen on it is very specific about it. And we have as greater diversity of antibodies in the range of 10 raised to power 16, and practically it is possible to have around 10 raised to power 9 antibodies. And this, as I told in the beginning, B cell can be stimulated in order to produce the antibodies. If you see the structure of the B cell, which is showing the presence of the antibodies on its surface, let's say it encounters a bacterium, for example, and these are the epitopes on the bacterial surface. On that, the paratope of the antibodies react with this. This leads to the multiplication and the proliferation in the B cell, and it will terminate into the plasma cell, which is eventually going to secrete the antibodies. So this is how it is going to happen. Again, there'll be a lot of things which will be happening in between. That is from the activation point of view, it gets certain signal from the T helper cell in order to secrete the antibodies and for its proliferation as well. That is not today's topic. Let's go into the structure. If you look at the antibodies, you'll find there'll be two light chain and there'll be two alpha chain. This is the light chain that is shown over here, and this is the heavy chain. If you look at the structure of the heavy chain, one thing you can easily make out, there'll be a lot of disulfide linkages between the chain and intra-chain as well. And this is the amino terminus, this is the carboxyl terminus. And if you see carefully, what you'll find is the light chain have the one variable region and one constant region. Whereas if you see carefully, you'll find the heavy chain will have the one variable region and there'll be three constant region. In between, they have the hinge. So they together joined with the disulfide linkages, and for the folding purposes also we require the disulfide linkages. So this variable region, in that also you have the hypervariable region which could bind with the antigen. So binding site to the antigen will be always present in the variable region. It has a distinction of a single amino acid. Even if the two antigen differs in the single amino acid structure, they will be recognized by the antibodies. And the name of the antibodies always be given based on the kind of heavy chain it has. It has the 5 different heavy chains: alpha, delta, epsilon, mu, and delta. On that basis, it has been given the name like IgA, IgM, IgD, IgE, and IgG. Similarly, the light chain, it is going to be either the kappa chain or it's going to be lambda chain. So if you go into the structure, this is also called as the immunoglobulin. based on the kind of protein it has. Now, if you see the heterodimers, as I told you in the beginning, and they together joined by the disulfide and certain other non-covalent linkages. Disulfide linkage is a kind of covalent linkage. Variable region will have the complement determining regions, CDR, or hypervariable region, and the constant region will have the function of the effector that will help the bind to the host cell receptor, and also it plays important role in the glycosylation. This structure makes the things very clear. You have the light chain and the heavy chain, and here the variable regions are shown and hinge is going to be there. Now we have the three families of the immunoglobulin gene exist, each on a separate chromosome. For example, kappa genes will be present on the chromosome number 2, lambda genes will be present on the chromosome number 22, and the heavy chain genes entirely present on the chromosome number 14. And each family consists of the series of the variable region genetically linked to the one or more C region. I told you already, constant region will determine what kind of antibodies it is going to end up with. And if you see the structure, this already we have seen, there'll be many ways to study this. For example, if you subject these antibodies, which is a glycoprotein, to be cut by the pepsin, we'll find the two region FAB prime 2, and we have the FC fragment. FC fragment is the crystallization fragment. Sometimes you get confused with this region because this region is going to bind with the complementary region as well. Complements, which are the set of 30 proteins which can bind to this. Again, if it is another proteases, if it is going to be cut, it will be broken into the FAB. There'll be two fragments, and the FC, there'll be another. And if you're using the beta mercaptoethanol, which is a disulfide linkage breaker, it will be further broken by into its individual chain, that is a two heavy chain and two light chain. So FAB, I told you, the antigen binding region, and FC, which is the fragment used for the crystallization, and the size, light chain is around 25 kD in size, and the heavy chain just double, 50 kD in size. This is the basic structure. If you look at, there'll be 5 chain is going to be there in the constant region, which is the part of the heavy chain. So the 3 of them, if you see carefully, will have the hinge. You can see over here, that is the gamma, delta, and alpha. That means it is going to make you the IgG, IgD, and IgA. The IgD, Ig is going to be the 3, and IgG and Ig epsilon, which is going to make this these the two chains will not be having the hinge present over there. So let's first talk about the IgM. As I told you in the beginning, in the IgM, the heavy chain is going to be the mu, and this is going to be the first response, because mainly it is present on the the B cell. So this is going to be first response to the antigen, but this is not going to cross the placenta. Then the IgD, which is required for the B cell activation. IgG is the most common one, where it is going to have a very important region. The heavy chain is going to be of the gamma, and this is one which can pass the placenta. So this is going to provide the passive immunity to the fetus. Then IgE, basically it is for the allergy purposes, found on the found associated with the mast cell and the basophil, because they're going to produce the histamine. IgA, which is the secreted form of the mucous membranes in colostrum, it is found in the tears. And these are the 5 chains, which is having some subclasses as well, like the IgG, which is having the gamma is the heavy chain. It may be found in the 4 subclasses, and the light chain could be only one, either the kappa or gamma, not both together. So this is how the structure is going to be formed. Similarly, for A, we have the 2 variants. So you can see IgG and IgA, they are having the subclasses, and other 3 will not have any classes. Now, if you see the IgG, 80% of the total serum, this is the one of the antibodies which is found in the maximum in concentration with the different biological activity. You can see the 4 subclasses, 1, 2, 3, and 4. IG1, 3, 4, they can easily cross the placenta, play an important role as a passive immunity, because these antibodies are produced by the mother and going to play important role in the fetus body. IG3 is the most effective complement activator, followed by the IG1, IG2, which is less efficient, and IG4 is not able to activate the complement at all. Basic reason is that, one which is able to form a dimer, and the distance between the 2 monomers which matters, and that is how it is going to activate the complement, which cannot fit into that, will not activate the complement. IG1 and IG3 bind with a high affinity to the Fc receptor on phagocytic cells and thus mediates the opsonization. It will cover up the any antigen or any organism like the bacterium, and IG4 will have the intermediate affinity for Fc receptor. IG2 has extremely low. That means IG1 and 3, they're having the high affinity to bind with the Fc region. Next one is the IgM, which is supposed, if you just count the number, there'll be 5 number. That is going to be a pentamer, and each will have the 2 binding sites. That means the valence is going to be 10. So it is going to be the 5 to 10% of the total immunoglobulin produced. And here we have the J chain, which will play an important role in the polymerization. So the pentameric structure and the decavalent in binding with the antigen. Then we have the IgA, is going to be 10 to 15% of the total immunoglobulin, and basically it is found in the the secretion part, or plays important role in the mucosal transport monomer, but sometimes it can make the polymer with the help of the J chain, and they have the poly Ig receptor, and they have the secretory component as well. This is how they're going to cross the, if you see the structure, this is a dimer IgA will the bind with the Ig receptor, and this is the epithelial cell, and once they'll be packaged into a vesicle, and once the vesicle is going to be fused, and here you can see this chain is going to be this part especially is going to be removed, and so that they can be secreted out. IgE, I told you, they play important role in the allergic reaction or maybe in the hypersensitivity reaction. They do the activate many secretions by the mast cell and the host macrophages, where they're going to release many chemicals or the granules, which in the process can activate or can bring about a lot of inflammatory reactions. Then the IgD, basically it is found bound to the mature B cell, and it is the least in the percentage. So this is the IgD first discovered when a patient developed a multiple myeloma. It is a kind of cancer, and here the myeloma proteins fail to react with the anti-isotypic antisera against the then known the isotypes, because the 3 only known. So when it was the 5th one which is bound to this, and this is the IgD, and has a serum concentration 30 microgram per ml, and constitute around 0.2% of the total immunoglobulin which are present. So it is going to be least in the number. IgD along with the IgM, they're the major membrane bound immunoglobulin expressed on the mature B cell. So you can see over here all the 5 antibodies shown over here, and I told you especially the hypervariable regions which play important role in binding with the antigen. The place where it is bind to the antigen is called as epitope, and the place on the antibody is called as paratope. There's a few definitions we need to understand. One is going to be isotype. Isotypic determinant or constant region, which determine that the collectively define each heavy chain classes and subclasses, and antibodies routinely used for the research purpose to determine the class or the subclass of the serum. Then we have the allotype. Although all members of the species inherit the same set of isotype genes, multiple alleles exist for the same gene, and these going to vary them in the allotype. So what exactly the allotype is? The sum of the individual allotypic determinant displayed by an antibody determinant and its allotype. In human being, if you look at, there'll be many. For example, the gamma chain allotypes is going to be G1M, G2M, G3. That there's so many that is going to represent the allotype. Then we have the idiotype. So especially what is the unique sequences of the amino acids found in the variable region of the heavy chain and the light chain that will determine the idiotype. So if you see carefully, you see the A is the isotypic determinant, like you see the mouse IG1 and mouse IgM, they have a difference. You can see over here. Then you can see the basically based on the kind of chain they're going to have in the constant region. Allotypic one is that you can see the IgG1 and IgG1 from the mouse, but they have a different binding site for the different antigens. And the idiotype you can see over here, mouse IgG1, and you can see they will bind to the different kind of antigen in this region. And if you see over here, they're going to bind in this particular region. So we have the three ways we can determine the antibodies. And as I told you in the beginning, the activated B cell will get the interleukin 2, 4, and 5. They get proliferated, and all they're going to develop into the, for example, if it come across with the interferon gamma and the TGF beta, so interferon gamma will lead to the IG2 and IG3, and TGF tumor growth factor beta will lead to the IgA and IG2B. Similarly, IL4 245 will lead to the Ig or IgG1 and the IgM. That means the kind of external exposure it gets, rather it is the cytokines, accordingly it is going to develop into. So this is the structure. Already we discussed about it. We have the heavy chain and the light chain. If you see this over here, so this is going to show you the beta structure, and these arrows show which way, whether they're the parallel or the anti-parallel. So similarly, you can see these structures are found in the both light as well as in the heavy chain. So light chain, this is the C domain, and the light chain, this is the V domain. And if you look at the hypervariable region, so what you'll find is that the few in the heavy variable region, and the light there'll be 3 CDR1, CDR2, and CDR3. Similarly, if you see the hypervariable region and the CDL, especially you see the CL, the constant region in the light chain, and this is the constant region in the variable region that is shown over here. And the epitope, as I told you, this place which is on the antigen, and the paratope, the place onto the antibodies, especially on the variable region of the light and the heavy chain. Similarly, the one experiment, that is a hybridoma technology, which got a Nobel Prize to Kohler and Milstein for this discovery, and this is playing a huge role in the health sector, that is a monoclonal antibodies. So this is how the hybridoma are going to be selected, the specific media we call the HAT, hypoxanthine, aminopterin, and thymidine, and this process is little different. So that is going to be this is how it is going to be developed, and there'll be a lot of clinical uses of the monoclonal antibodies. For example, the diagnostic purposes, pregnancy, pathogen, blood level of the drugs, tumor antigens, imaging, tumor metastasis, therapeutic reagent like the immunotoxin, and can be used in the enzyme industry because of the specificity of this region. So antibody plus enzyme. So antibody part will play important role in recognition, and enzyme is going to cleave it. And these are the many ways the antibody diversity, huge number of the antibody diversity can be developed. For example, the multiple germline genes, then the VDJ joining, junctional flexibility, palindrome, nucleotide addition, P and N addition, somatic hypermutation, and combinatorial association of the light and heavy chain. This leads to the huge diversity in the antibody structure. So hopefully this lecture is going to help you. Do subscribe, watch, and share with others as well, so that you can get the maximum benefit of my lectures. Thanks a lot.